Okay, the title of my presentation is Innate Immunity, Soldiers or Sentinels, the case of NK-reg cells in MS. So I will start with the concept of innate immune cells. In the past, innate cells were considered classically as stupid soldiers that, like first-line soldiers, would kill the enemy, and prepare the scene as adaptive cells, more evolved one, would prepare to combat. But in last years, this concept has changed a lot, and innate immune cells are now regarded as not only first-line soldiers, but also sentinels who are able to interact with the generals, let's say, which are the adaptive cells, and influence their outcome. So I will start with a brief overview of multiple sclerosis, which is the disease which is the focus of my presentation. So MS is an autoimmune disease, an immune-mediated disease of the central nervous system. It is a multifactorial disease. Different environmental factors act in genetically predisposed individuals to promote an autoimmune response attack against myelin antigens. Clinically, multiple sclerosis is characterized by two phases of disease. The disease usually starts as a relapsing remitting disease, where patients usually have a low disability. Okay, is the microphone okay? Yeah. And is characterized, this relapsing remitting phase is characterized by flares of inflammation that go from the peripheral blood to the central nervous system to mediate, to attack the myelin and mediate the relapses. Over time, inflammation fades and a neurodegenerative phase starts, which is characterized by not only by inflammation, but also by degeneration of the central nervous system. So this is a cartoon summarizing what happens in MS. There are, at the beginning of the disease, there are peripheral lymphocytes. Usually, so MS is characterized as a T-cell mediated disease. So T cells are primed in periphery by unknown triggers, enter past the blood-brain barrier, enter the CNS, and mediate inflammation. Inflammation causes demyelination, and over time, demyelination results in the neurodegeneration. And these are just some macroscopic brain views of the brains of patients with progressive advanced MS that show the demyelination. The white matter is not white anymore in different areas, and a big atrophy, so which is the result of the neurodegeneration. So neurodegeneration is visible also as transected axons. So the demyelinated axons over time die. As I said before, MS is considered as a T-cell mediated disease, and this is proven by pathological and CSF studies and by animal models that show that vaccination with the myelin antigens promotes a T-cell mediated attack of myelin. And so what are the cells that are, the T cells that are mediating the damage in MS? Mostly probably the T helper 1 and T helper 17 cells, with a theory that says that maybe Th17 are the cells that enter the CNS at the beginning of disease, and then start the inflammatory process that recruits later Th1 cells. We all have autoreactive cells in our peripheral blood, but most of us luckily don't have MS. So why does MS start in some individuals and doesn't start in others? One possible explanation, or a part of the explanation, is in defects in controlling the activation of autoreactive T cells. So we all have autoreactive T cells, but we also have regulatory cells that control T cell activation. And in MS are more regulatory subsets that are more characterized are the FOXP3 Treg cells and the TR1 T regulatory 1 cells. And there are several studies that have shown, mainly by the laboratory of David Hafler, formerly in Harvard and now in Yale, showed that a defect in regulatory T cells is associated with multiple sclerosis. And also David Hafler showed also that Tregs, he showed previously that there are problems in the number of Tregs in MS, but more recently, this is 2012, I think, he showed that the Tregs in MS people who have an increased levels of IL-12, interleukin 12, which is a pro-inflammatory cytokine in peripheral blood, Tregs become less functional and become like pro-inflammatory cells, producing higher levels of interferon gamma and pro-inflammatory cytokines. So let's go back to the innate immune system. What is the role of the innate immune system in this picture? So the innate immune system is more phylogenetically ancient than the adaptive immune system. Now, this is a nice review on the phylogeny of the immune system and the interactions between the adaptive and the innate immune system. That says that the modern view is that the outcome of T cells and B cells responses depends on the influence of innate immune cells on innate immune cells on those adaptive cells. And here is a cartoon showing that the adaptive immune system developed later in evolution, but all those more primitive organisms have an immune system and are able to recognize external antigens. How do they do that? They do it through the innate immune system. And this is another way of showing the same thing, that primitive organisms don't have all those adaptive immunity, TCR rearrangement, and so forth, so on, but they have the innate immune system. To work, the innate immune system must be able to recognize foreign antigens. Since innate cells do not have the TCR and the BCR, so receptors that characterize the adaptive immune cells and are able to recognize foreign antigens, how do they do that? So it was Genoa, the famous immunologist, that is also the author of a famous immunology manual, who described who had the idea, the vision of how innate immune cells could be able to recognize the antigens. And his vision, which was published in 1989, later became confirmed. So he described the three mechanisms that innate immune cells use to recognize antigens, foreign antigens. The first one is the known self. Innate cells are able to recognize the known self. The second one is the aberrant self. And the third one is the missing self. So these three mechanisms are used by innate cells to recognize foreign antigens. And this is a less funny picture of showing this mechanism. So the main way used by innate cells to recognize the known self is using specific receptors like the famous toll-like receptors, while aberrant self and missing selves are recognized through the modulation of molecules in the surface of self-diseased cells. So, and here we go to the NK cells. The NK cells are innate immune cells that use these mechanisms to recognize diseased cells, cancer cells, or infected cells. The main and the most probably bestly described mechanisms they use to recognize foreign cells is the missing self mechanism, which is that NK cells do not kill cells that express self cells and the self molecule. And the self molecule that all of our cells express are the HLA class 1 molecules. So if an NK cell meets a healthy self cells which expresses the MHC class 1 cells, this binds to an inhibitory receptor that blocks the NK cells, prevents the NK cells to attack the foreign cells. So this is the missing self mechanism. But so when a cell is attacked by a virus or becomes neoplastic, it downregulates the expression of MHC class 1 cells, and so the NK cells can kill it because they don't find the stop signal, so they kill. This is not so simple. This is a simplified view. NK cells also have activating receptors, similar to the toll-like receptors in concept, and so it is the balance between the inhibitory and activating receptors that tells the NK cells whether to kill or not. NK cells have been as a part of the innate immune cells, NK-like receptors are expressed in very primitive organisms like this protochordate C organism, and also in other zebrafish, for instance, organisms. These zebrafish cells, some zebrafish cells in particular, express some receptors that are similar to the killer immunoglobulin-like receptors expressed by NK cells. And this graph shows that these receptors are in some ways similar in some domains to the T-cell receptors. The killer immunoglobulin-like receptor are prototypical inhibitory receptor of NK cells that bind to MHC class 1 molecules. So in humans, NK cells are broadly characterized as lymphocytes, not expressing the CD3, which is a marker of T cells, and expressing the CD56 molecule. CD56, also called neural adhesion molecule, NCAM1, is typical of the human NK cells. So in animals, it's not expressed, and especially, and this makes it not easy to correlate the role of NK cells and NK cell subsets in humans and in animals, and to use animals as a model for disease. The NK cells were recently recategorized as a part of the innate lymphoid cells family. As you might know, there is this big group of the ILCs, innate lymphoid cells, that are cells belonging to innate immune system and mirror in some way the T helper cells. So these are adaptive cells, these are innate cells. So there are ILC1 cells that are similar to the Th1 cells, group 1, group 2 that are similar to the Th2, and group 3 that are similar to Th17, Th22. NK cells are cytotoxic in innate lymphoid cells belonging to the group 1. The group 1 ILC1s also includes non-cytotoxic cells that I won't go through. Okay. So NK cells fully fulfill what I said before of innate cells as not only being soldiers but also sentinel, because they are not only able to kill infected cells or diseased cells. In particular, NK cells in humans can be divided into two main subsets based on the expression of the CD56 marker. This is peripheral blood. This is a real plot from our laboratory that shows that there is the big group of NK cells expressing lower levels of CD56 and a small group of CD56 bright NK cells. This subset were described in 2001 firstly as immunoregulatory cells. Why? Because they were able to release, compared to the other, the CD56 NK cells, they were able to release higher amounts of cytokines, both pro-inflammatory like interferon gamma and anti-inflammatory like IL-10. So the white boxes are the CD56 DIM and the black are the CD56 bright NK cells. And you can see that with different stimuli, the CD56 bright release more, much bigger amounts of cytokines than the DIM. And here, these plots also show how they look like in terms of the expression of, it's not written, of CD16, which is another prototypical NK marker. It is the receptor of the Fc fragment. CD16 is expressed by all CD56 DIM NK cells, but only by a part of CD56 bright NK cells. And in subsequent years, it became clear that CD56 bright, through release of cytokines, interact with other innate cells, like antigen-presenting cells, and these cartoon shows that they produce interferon gamma, which activates APCs, but are also able to respond to adaptive-derived cytokines like interleukin-2 that activate these cells. And it's very important to say that these cells are the majority of NK cells in lymph nodes. So while in peripheral blood they are just about 5% of circulating NK cells, so a very small number of total PBMCs, in lymph nodes, those cells are the vast majority of NK cells. And we know that in lymph nodes is a crucial place where the recognition of antigens and presentation of antigens from APCs to adaptive cells occurs. And this, okay. So and now I'm going to some results of our laboratory. We asked ourselves whether the CD56 bright NK cells were truly immunoregulatory. So does this secretion of cytokines result in some changes in the adaptive immune response? We did a study with IL-27, which is an immunoregulatory cytokine, which is released by APCs. And we found that when we culture the CD56 bright NK cells with this immunoregulatory cytokine, they become able to suppress the proliferation of autologous T-cells. These T-cells, so this is the suppression of T-cells alone, so by definition it's 0, and these are CD56 bright NK cells cultured with IL-27. And this was associated with the increased production of perforin and decreased and abolished in transwell, suggesting that this suppression was contact dependent and might have involved a direct cytotoxicity. Since those cells are known, classically described as producers of cytokines, this could be a bit strange because the other subset, the CD56 DIM, is the truly cytotoxic one, while these cells were traditionally considered as less cytotoxic. And so what about these cells and multiple sclerosis? These cells gained attention in the field of MS with a trial assessing the effect of Daclizumab, which is an anti-CD25 monoclonal antibody in MS. So Daclizumab was initially developed with the idea to counteract the activation of T cells. CD25 is part of the high affinity IL-2 receptor. So the idea was, the initial concept was to dampen T-cell mediated inflammation. And of note, the trial showing the efficacy of Daclizumab in decreasing the relapse rate in MS has been published last month. So in this was, I think, 2006, I'm not sure. But while Daclizumab was developed, the group of Roland Martin studied the possible mechanism of action. And surprisingly, they didn't find a decreased, a direct effect on T cells of Daclizumab. Rather, they found an increase in the number of circulating CD56 bright NK cells. And the increase in CD56 bright NK cells was inversely correlated with the activity of disease at MRI. And so they also found that CD56 bright NK cells from patients enrolled in the Daclizumab trial became able to kill autologous T cells. The same group performed many studies on this therapeutic setting. They characterized the effect of Daclizumab, showing that what happens is that since T cells, due to the downregulation of CD25, T cells do not respond to IL-2 anymore. But so there is more available IL-2, which binds to the CD56 bright NK cells through the intermediate affinity receptor. So while T cells don't respond to with the intermediate affinity receptor, the CD56 bright NK cells use it in order to respond to IL-2. So treatment leads to increased proliferation of CD56 bright NK cells. And CD56 bright NK cells of patients treated with Daclizumab become able to kill autologous T cells through transfer of a killing molecule, a lytic enzyme, which is called granzyme K. You might know that one way that NK cells use to kill is to make holes in the membrane of the target cells through perforin, and then to send inside the lytic enzyme like granzymes. So this granzyme K is increased in cells treated with in CD56 bright NK cells in treated patients, and is transferred to T cells. So this is very important because in MS, as you might know, we don't know the mechanism of action of many treatments, except for few of them. Most treatments probably have a they have a very unexplained mechanism of action. While for Daclizumab, it seems there is a real biomarker, so the treatment increases these cells, and this is correlated to killing of autologous T cells and disease remission. And the same group also described, but this is a bit, it's a bit not easy to read, described that Daclizumab decreases the number of bad innate lymphoid cells by transforming them in CD56 bright NK cells. Okay, so summarizing what we know, what we said so far, is that NK cells can be divided in two subsets. The CD56 bright NK cells are very interesting because they are the majority of NK cells in lymph nodes. They produce the cytokines. They increased in patients treated with Daclizumab. And for the sake of synthesis, I didn't talk about other settings, but CD56 bright NK cells were described to increase in pregnant MS patients, and pregnancy is associated to disease remission, as well as in patients treated with interferon beta. These are small studies, so I think the Daclizumab studies are more important. So we started from here to ask what is the role of these cells in immune regulation, and whether these cells are in some ways impaired in untreated MS patients. Because we know that Daclizumab makes them cytotoxic, but what's in untreated patients? What is the status of untreated patients? Do their CD56 bright NK cells function well? Are they able to regulate the T cells? And so this is what we did, and this is the final part of my talk. We wanted to investigate whether CD56 bright NK cells acquire an immunoregulatory function in healthy subjects, and whether this is impaired in multiple sclerosis. Experimental design is that we purified the two subsets from total NK cells derived from peripheral blood of healthy controls at the beginning, and also MS patients later on. We pre-activated them with pro-inflammatory cytokines, IL-12 and IL-15, and I'm going to explain why. We resorted living cells, and we cultured them after this pre-activation with autologous T cells in presence of T-cell specific stimulus, which is anti-CD3, anti-CD28 beads. And we also, this is what, in general, what we did. And why did we choose IL-12 and IL-15? First of all, because they are prototypical cytokines that are well known for activating the NK cells. And second, because these cells, as I also said before, these cytokines are described as increased in MS patients. APCs from MS patients produce more IL-12 and IL-15. So this was, for us, a setting that could mimic what happens in the blood of our patients. So I will go through all of this. First of all, we assessed whether CD56 bright NK cells and the CD56 dim NK cells, the other subset, were able to suppress autologous T-cell proliferation upon this stimulus. And we found that CD56 bright NK cells effectively suppress, in healthy subjects, effectively suppress the proliferation of autologous T cells. We wanted to assess what was the mechanism of this suppression. We assessed the death of CD4 T cells after culture with CD56 bright NK cells, or CD56 dim, and we found that suppression was associated to increase the death of CD4 T cells by FACS, by flow cytometry. We checked the degranulation of CD56 bright NK cells in presence of T cells. Degranulation is an indirect way to measure cytotoxicity of NK cells, because there is an assay which is called the CD107 assay, which measures the expression, the surface expression of CD107 as a marker of degranulation of the cells and killing. And we found that CD56 bright at a random time point were able to degranulate more than CD56 dim NK cells. This is a very low level of degranulation, but it's important to say that we cultured the cells together for 5 days, and we randomly chose one time point for degranulation because of the low number of cells. The degranulation of CD56 bright NK cells towards T cells was almost zero when the T cells were not activated. and when T cells and NK cells were not in contact, suggesting that CD56 in general, that CD56 bright NK cells kill autologous T cells, autologous activated T cells, but not non-activated T cells. We asked ourselves why did the NK cells kill the T cells? Because T cells are self cells, and since they express HLA class I molecules, they should be not targeted by NK cells. We tried to, so this is the classical suppression assay. We performed it while blocking different activating receptor expressed on NK cells or the HLA class I in T cells. And we found that when we blocked with a mix of antibodies a group of receptors called the natural cytotoxicity receptors, suppression went down. And we found that the natural cytotoxicity receptors involved in this downregulation were the NKp30 and NKp46. So these receptors are part of the NCR family and are and recognize mostly unknown self ligands together with known self ligands. And these receptors are expressed by the CD56 bright NK cells in presence of inflammation. How would the NK cells kill the T cells? So first, by real-time PCR, we found that the activation with IL-12 and IL-15 increases the transcription of granzyme B, which is one of those lytic enzymes that the NK cells use to kill the target cells. By intracellular staining, we found that the content of granzyme B in T cells, when cultured in presence of CD56 bright NK cells, increased compared to when the CD4 T cells were alone. Parallelly, the content of granzyme B in NK cells decreased as they were cultured with the T cells, suggesting that the granzyme is transferred from the NK cells to the T cells. And accordingly, inhibiting the effect of granzyme B on target cells decreased the suppression of CD56 bright NK cells. So, so far, we showed that CD56 bright NK cells in healthy subjects are able to control T cell activation by direct cytotoxicity that involves the natural cytotoxicity receptors and the release of granzyme B. We then asked ourselves what was the function of these cells in untreated multiple sclerosis. So the number of these so-called NK regs was similar over total NK cells, was similar in MS patients compared to HCs, healthy controls. So there is not a defect in the number of these cells. And I remember, I say again that Daclizumab increases their number, but it's not defective at the starting point. But what we observed in a very reproducible way was that the CD56 bright NK cells from MS patients are less able to suppress the proliferation of autologous T cells, autologous. So why do CD56 bright NK cells from MS patients function less? First of all, we evaluated whether we were not dealing with CD56 bright NK cells. And this was, since we sorted cells only based on the expression of CD56, it could be possible that activated CD56 dim NK cells, which upregulate the CD56, but are not real CD56 bright NK cells, are just activated CD56 dim, would fall in our sorting gate. And so we were not just dealing with the right cell population. So we characterized the cells from MS patients and healthy controls through multiple markers that distinguish the CD56 bright from the CD56 dim. And we did not find any differences in our patients. So the CD56 bright, they were really CD56 bright, albeit not functional. Then we evaluated whether these cells would not work well because they did not express enough of the activating receptors or would not release enough granzyme B. So we found actually that the expression of the receptors, and the MS patients are blue, this is an example and these are the overall data, was slightly increased in MS patients rather than decreased. I'm not sure whether these have as a clinical significance, but could also represent a kind of way of counteracting the defective function. And the expression of granzyme B in healthy control, in CD56 bright NK cells of healthy control versus MS patients was not different. So at this point, we finally asked whether it could be a problem of the T cells, which would not respond to the regulatory effect of the NK cells. And what did we found? So these are the healthy controls, and these are the MS patients. And here we show what happens when we block the HLA class 1 in the T cells. This is also a part of what I've shown before, blocking receptors in a suppression assay. So this is suppression, and when we block the HLA class 1 in T cells, suppression increases, but not in a significant way. In MS, suppression is much lower at baseline. When we block HLA class 1 in T cells, it jumps up at more or less the healthy controls level. And accordingly, we assess the expression of the HLA class 1, which is recognized by the CD56 bright NK cells, which is the HLA-E molecule, a very a molecule which doesn't have much variability and is very similar in all of us. And we found that the T cells from MS patients express higher levels of this inhibitory signal molecule. So this is in this cartoon, this is just a summary of what could how our experiments could explain what's happening in MS. So in a healthy control, and this is a cartoon showing a lymph node, and this is just theoretical because all our experiments were done with ex vivo isolated NK cells from peripheral blood. So in lymph nodes, the APCs present myelin antigens to autoreactive T cells and release pro-inflammatory cytokines. These pro-inflammatory cytokines, together with activating the T cells, activated the CD56 bright NK cells that control the proliferation of T cells through granzyme B. In MS, the increased expression of HLA-E by T cells blocks the control by CD56 bright NK cells and makes autoreactive T cells proliferating. And this is unpublished, and so we are submitting it now. And of course, this does not explain the whole pathogenesis of MS. But our hypothesis is that also defects in innate immune mediated regulation of the adaptive response coexist with the defects in adaptive regulating responses to mediate MS phenotype. I'm not sure whether this is, probably this is an initial part of the pathogenesis rather than a later stage, since the inflammation characterizes more the early phases of multiple sclerosis. And this project was found by Italian MS Society and was I started this in Boston and was carried out partially in collaboration with Harvard University. A very important collaboration came from the group of Professor Alessandro Moretta, and particularly Emanuela Marcenaro and Simona Sivori, which are experts in NK cell biology. Thank you.